So one of my most profound memories back when I was little was watching David Attenborough's nature documentary, The Private Life of Plants. It was just incredible to see how diverse plants were and how every single plant was characteristically unique. I'm Dennis, I'm the founder of Living Glass, and today we're at the University of Guelph where we'll show you how we make our products.
So plant tissue culture is a technique where we take a small piece of tissue from a plant and culture it in a very controlled environment. And so what we do is we provide carbon sources for them instead of relying on photosynthesis. So in a natural setting, a plant uses photosynthesis to convert sun energy to fix carbon from the air, from carbon dioxide, and turns it into sugar. And so what we're doing here is we're just providing the sugar so that the plant doesn't have to do that. And by putting it into very controlled conditions, we can manipulate the light intensity, we can manipulate the carbohydrate source, the nutrient source, and all of the other parameters, plant hormones, for example. And what this allows us to do is actually direct the plant growth however we want. From the conservation angle, what this allows us to do is take a very small piece of a plant from the wild. We can multiply it into thousands of plants without impacting the wild population. And then we can use those plants to either put them back into the wild, we can maintain them in a botanical garden, we can cryopreserve them in liquid nitrogen. And basically what it does is it gives us a reservoir of that specific species. A good example of a success story would be golden paintbrush. And this is an endemic species to the Pacific Northwest, including southern British Columbia, Washington State, and a bit of Oregon. And there's only 11 populations left in the wild. So what we were fortunate enough to get is 4 plants. So they gave us 4 plants from British Columbia. And from those 4 plants, we were able to make thousands of plants, and we were able to cryopreserve them and then send them back into the wild. And so this is a good example just to show that we can take the plants, bring them into the lab, and we can keep them in the lab as long as we want, and then we can reintroduce them. And so by reintroducing them, what this means is that if they happen to go extinct in the wild, we have a backup population in the lab that we can use to replenish that population.
In high school, my curiosity of plants got me into plant collecting. And so I had this elaborate setup with grow lights and terrariums that I would grow hundreds of very unique plants in. These are plants that you couldn't find at your local nurseries because they're either too rare or too difficult to commercialize. So my first company I founded was focused on selling these collector-level plants, but I always felt bad because not everybody could purchase my plants. You needed to have dedicated grow spaces and you needed to have space. So in 2018, I founded Living Glass, and we empower individuals to grow unique plants with minimal care. We use a method of growing plants called tissue culture, which uses nutrient gels to support the metabolic needs of plants. So today we're going to show you exactly how we make our tissue culture products. So let's go. So today we're going to be making a custom Living Glass order. The same processes that we use in tissue culture is applied for making Living Glass plants. What we have here is sugar, a gelling agent, and a micronutrient blend. So all of these things will go into our beaker here and it'll get mixed up and heated. To fine-tune our solution, we'll test the pH of it. Typically our media for carnivorous plants is very acidic. So we use sodium hydroxide, which is a base, to neutralize that acidity. So now the final step is just adding in the gelling agent. So once the solution is at a boil, we can go ahead and pour it into our glassware. So now we'll bring this into our autoclaving room. So over here we have the autoclave room. As the name implies, we have these 2 big machines here called autoclaves. What they do is they heat up the solutions and all our equipment up to 121 degrees Celsius. And this is very important for sterilizing all our tools and preventing any contaminants like bacteria and mold from growing in our cultures. So what I'll do now is I'll take aluminum foil and I'll just wrap the top of our vessel to make sure once we take it out that no contaminants are going to get back in. This tape here is called indicator tape, so it lets us know if the proper temperatures have been achieved. Typically we'll allow this to run for about 30 minutes, and this gives us sufficient time to kill off all sorts of contamination. So now we'll just wait for a bit, and then afterwards we'll unload the solution. So this solution, it's just been heated and sterilized, and as you can see by the indicator tape, this sterilization was successful. And so now we'll bring this into our laminar flow hood, which is an environment that allows us to transfer plant materials without the interference of contaminants. And we'll let this cool before we start putting plants in. Over here, we have our main growth chamber. So this growth chamber here is optimized to maximize the growth of our plants. So yeah, let's take a look. So being in this room, I definitely feel like a kid in the candy store. In this tiny amount of space, believe it or not, we could grow hundreds of different kinds of plants. We have sundews, we have pitcher plants, and we even have Venus flytraps. So let's take a few of them out and take a closer look. So right here, we have something called a laminar flow hood. What the laminar flow hood does is it sucks in air from the external environment and passes it through a HEPA filter, which filters out particles up to 0.3 microns. And this is very important because it helps us maintain a sterile environment while we transfer plants. So before I put everything in this laminar flow hood, what I'll do is I have a solution of 70% ethanol, alcohol, and I'll spray everything down. And as you can see, all of these things have been autoclaved, so they are sterile. So once our gels have cooled and solidified, we can begin the transplantation process into our vessel. All of this has to be done in a laminar flow hood to prevent contamination from pathogens during the transfer of plant material. What we have here is a type of plant called a Threadleaf Sundew. They have these long cylindrical leaves that are covered in sticky hairs. And if you haven't already guessed, they use these leaves to trap and digest insects. They are a native in Canada and actually considered endangered because of habitat loss which stems from commercial peat mining. We'll take a look at the plant and see if there's a growth point, and I'll break it from the clump. And now we'll go ahead and simply plant it into the gel. So now we've pretty much finished the process of tissue culture, and all that's left for us to do is to put it under some LED lights and wait for it to grow. Tissue culture is amazing because not only does it allow us to mass propagate plants that otherwise would grow very slowly, but the plants grown in this method are also extremely easy to keep. There's many different species that people don't even know exist and might not ever see in person. At Living Glass, our goal isn't just to deliver on some cool looking houseplants. Our hope is that we can also spread some awareness on species that are diminishing in the wild.